Today we're joined by Sara Isbell, CEO and co-founder of Enabled Therapeutics, a precision neurology company aiming to transform how we treat brain diseases. With over 22 years of experience in neuroscience and drug development, Sara helped discover Capton, a revolutionary drug technology that activates only at disease sites, offering targeted disease-modifying treatments for conditions like Alzheimer's, epilepsy, and glioblastoma. Under her leadership, Enabled Therapeutics is advancing a bold pipeline of therapies that promise safer, more effective outcomes by tackling the root causes of neurological disorders with unprecedented precision. Now, Sara, thank you so much for joining us today. And first question, can you tell us about the founding vision behind Enabled Therapeutics and what inspired the development of Capton technology?
Well, first, thank you. That is great to be here. So Enabled Therapeutics was formed to take this technology that we discovered at another company forward. And the discovery was actually an accident. So it's not like a story, where we had this vision and we wanted to go out and treat Alzheimer's and all that. In actual fact, that's kind of a scary thing to do because it's a very risky area. But we had this discovery in 2010, so a long time ago, 15 years back, and we noticed something very different happening with our molecules that we happen to be working with. We were looking at a bunch of small molecular thiol molecules, and we thought we knew everything about them because we had run all these tests and we knew, what they could do in the body, how long they stayed, everything. And then we decided to put them on some neurons that we just happened to have in our lab. And we noticed that one of these molecules was rescuing the cells when we put them under oxidative stress. And we were at a company at the time, Raptor Pharmaceuticals, where they allowed us as scientists to follow the data, which is very rare. It's very, very rare these days. Usually the company has a strategy, has a plan, and you kind of have to stick to that. Well, the company was looking at cystinosis, which is, it's an orphan disease. It's a lysosomal storage disease, nothing to do with, neuroscience. But we had this discovery of how this one molecule was acting different in rescuing neurons in our lab. So we were allowed to follow that. And 6 years later, after following it, we formed a hypothesis in 2016. And we had the hypothesis that we think what's happening is that molecule is actually a piece of a bigger molecule that is being synthesized. It's being made by the damage. And when that molecule becomes something else, it's termed as oxidized, it becomes a sulfonate from a thiol. It was actually a known neuroprotectant in the literature. So we formed this hypothesis in August of 2016, and 1 month later, we are bought by a commercial company who wants nothing to do with early research. They bought us for our commercial. We had a drug out for cystinosis, which was a small thiol, and we were trying to make a better generation of these thiols. So when you see there's a chance to change neurology completely, I mean, if you can have the drug synthesized on site, we knew, being, with a neuro background, I knew if this was true, then we've changed neuroscience. We can now treat diseases that we could never treat before. If it was true, it was a far-fetched, but you have to follow up on something like that as a scientist. So we formed a company. We had to spin off because this commercial company bought us and said, if you want to take that and try it, we'll license it back to you. So we take this little discovery and hypothesis and form a company, raise some seed money, and we do all the tests and come forward now to 2025. We actually have proven it to be true. There is a way that you can synthesize drugs now on site, and the damage will do the synthesis. And it's been an amazing journey to get this far.
It really is, and a fortuitous set of circumstances. Had the circumstances been different, the outcomes would have been different too, surely.
Exactly. It's very serendipitous how this all came together. Incredibly. All the
Remarkable.
pieces.
Uh,
Yeah.
an excellent story. So now on to the specific conditions that we're going to be talking about today. So, Sarah, what makes neurological diseases like Alzheimer's, epilepsy, and glioblastoma, which is a particularly aggressive form of brain cancer, so difficult to treat with current therapies?
So there's a lot of reasons, but there's two main ones that's been very hard in the field. And the one you've probably heard of is the blood-brain barrier. Um, it's very hard to get drugs into the brain. But once you do, and there are ways to get around it, you can even inject. Once you do, you are actually now you're treating the entire brain. And the way you treat these conditions, because a lot is known about the pathology of Alzheimer's, of glioblastoma, of epilepsy. A lot is known. And we know, you got to stop the neuron firing, or you have to stop the immune system invading. These are ways you can do that. So if you can get through the blood-brain barrier, you're now inside. You're now going to stop the entire brain from firing. It's why epilepsy, drugs that do treat epilepsy make you very tired. The people are unable to operate vehicles. They can't drive. They can't go to school. It's a terrible condition to have because to treat it means you're sleeping all the time. You're treating the entire brain. So this idea of global activity of the drug is what makes it very hard to get around. For Alzheimer's, um, inflammation is a key driving force, and they know ways to stop the immune system invading the brain. But when you do that, you now develop viral infections because 99% of the population on the planet have a virus in their brain, probably dengue virus or JC virus. And our brains are constantly keeping them in check. If you remove that check, you now get meningitis, brain inflammation. So all these therapies to try to treat Alzheimer's or epilepsy come with these huge side effects, these adverse events that in a lot of cases are just too great to get to the market. When you're treating other body parts, such as, if you're going for cholesterol or, kidney disease, you do have side effects. You get cardiac toxicity and all that, but the body can manage a lot of the times. And what you're treating is worse than your side effect. But in the case of the brain, to treat these conditions, a lot of times the side effect is death. So it's been a huge challenge. One, with the blood-brain barrier, and two, you're treating the entire brain.
Indeed. So, a groundbreaking treatment in the true sense of the word, and that nothing even close to this existed before. Correct?
Correct. Correct.
Outstanding. So, next question. Enabled Therapeutics uses the phrase rewriting what's possible in neurology. What does that mean to you and your team?
It literally means rewriting what's possible, literally rewriting a script. So right now, if you go into the hospital and you get diagnosed with early stage Alzheimer's, they're going to write you a prescription for something that'll treat the symptoms. And they're also going to say, we're sorry. You're going to hear that a lot. We're sorry. There's nothing we can do. For these conditions, if you get diagnosed with it, you may be able to get drugs that make you a bit more comfortable, maybe extend your life by a few months. But really, right now, it's told you're sorry. But when a Capton, once we have our approach out there, it's going to be the script is now going to be take two of these twice a day, and and that's it by mouth. Um, there will be no more sorries. It will be here. Here's an oral medication you can have, and your Alzheimer's you will improve and get better. That is amazing. That is completely different than anything we have right now.
Amazing. And you sort of outlined some of the challenges with traditional CNS drugs, but probably worth revisiting again. So, what are some of the biggest challenges facing traditional CNS drugs, and why have so many failed to deliver meaningful results for patients? So, what were you up against essentially when developing a new drug, or indeed, what challenges did you look to remedy?
So again, when we fell into this, it was an accidental discovery. And what we discovered is that we found a way to get around every challenge. And that's what so we don't have a challenge. I mean, that's what we found is a way around the challenges. And the challenges, again, are twofold. Besides knowing what to target, when you're going to treat a disease, you got to know, okay, what's causing the disease? That for a lot of these conditions in the brain, we don't know. But what they have found out is ways to stop the neuron firing in a seizure or ways to stop the immune system from invading and causing swelling. They do know how to do these things, but then it's how do you get it across the brain? Well, when we realized when we formed our hypothesis was we're looking at a way you don't have to worry about the blood-brain barrier because we're being made on site. We never have to cross it. The drug is being synthesized in the damage. The damage is supplying the necessary chemistry to finish synthesizing the drug. And the partial drug we give is globally distributed, is benign. It's in very small amounts. It crosses membranes very freely. It will be evenly distributed in the body, being a thiol. So it was like we had this thought all at once, instantly, when we realized what we could be looking at. It solves the biggest challenge of the blood-brain barrier. We're in there. And then the second biggest challenge of the global activity. These targets we find that you can go for GABA, it's a receptor on immune cells, and it will stop them from migrating, from moving. So you can target that, but you don't want to do it to the whole brain where, it's keeping the virus in check. But imagine if you're making it on site because the levels of inflammation you get for Alzheimer's is way higher than what it's doing to keeping viruses in check. We don't mess with anything at the level of keeping things in check. But when you get extremely high inflammation like you do in MS or ALS or Alzheimer's, if you could synthesize the drug inside that and you're treating just that, now you get around that issue of affecting that healthy tissue. You're very localized. This is extreme precision we're talking about. So it really gets around all the challenges. And then there's another challenge they talk about, and that's translation. Things that work in the rodent doesn't work in man. And in fact, there was a paper done that found, and that has to do with the blood-brain barrier. They found that a lot of things, if you even just pet a rodent or talk to the rodent, its brain will become permeable. Something happens to it, and it actually, so all these studies that were done on rodents where it worked, it didn't translate in man because, it's not the same physiology. What we're talking about is chemistry. There's no biologics involved in our approach. It's solely the oxidation of a thiol group, meaning there is translation. If it's happening at the bench, it's happening in bacteria, it happens in the rodents, it will happen in man. We can say with 100% confidence that what we see in our rodent studies will translate into the human studies as well. So it solves all the major challenges all at once. It's beautiful. Beautiful, isn't it? Like the
It
discovery of
really
DNA.
is. No,
Yeah,
truly
yeah.
remarkable. Truly
And it's
remarkable.
nature.
And
It's nature.
I read something recently about inflammation linked to Alzheimer's or as a factor of Alzheimer's, but I think it's not a widely known fact. So can you explain the link between the two?
Yeah. Well, they find that the so that you hear a lot about amyloid. There's a lot of amyloid buildup, and amyloid is more like the scarring. It's like a scar. It's happening because of it. It's not the cause of it. But in addition to amyloid, there's a lot of inflammation because the body's trying to fix itself. So whatever the underlying cause of Alzheimer's is, and it's termed Alzheimer's because of the location in the brain. If you get degeneration in the motor cortex and stuff, it's Parkinson's. If you get degeneration of the neurons in your hippocampus, um, then they call it Alzheimer's. So it's the learning and memory area. So when you get that, and if it's genetic, then it's familial Alzheimer's. So there's a large umbrella of what's classified as Alzheimer's. Is it just dementia, or is it hereditary, or is it something? But in all cases, you do have the neurons are dying. You're getting the scarring of this amyloid, and the body's responding. The brain's like, oh, well, let's send the immune system. Let's send everybody to help clean this up. But you're sending a lot of cells. All these cells are coming to the site. They themselves are triggering damage, causing more cells to come. And you get this runaway effect of the immune system. And our immune system that's there to help us has actually now become pathological. Now, it's not known what will happen once if you could stop the inflammation with Alzheimer's because no one's ever done it. But what we do know is the tissue in these areas is highly inflamed, and that inflammation is causing this runaway effect of progression. More neurons are dying, causing bigger scarring, bigger holes. You're losing more memory. So if you could stop that from happening, the other thing that we'll find, if you don't have high inflammation, stem cells will actually settle and become neurons. Otherwise, stem cells are coming to the place. They see there's a lot of inflammation, and they too become a microglial cell, an immune cell, because they want to help out in the fight. It's your body trying to help, but it's helping too much. But if you could stop that, and then the stem cells come and turn into neurons, you can maybe even reverse the effects of Alzheimer's by treating the inflammation. Now, what is really causing it in the first place isn't known, but maybe we'd figure it out if we could, take care of the inflammation part, start repairing. That I'm sure, it always does, leads to more discoveries and more understanding about the disease.
Why is systemic toxicity such a major issue in neurodegenerative disease treatments, and how does Enabled address this problem? So, is there more to add in in that regard?
There's a little bit more to add. One thing again, so as I mentioned, all drugs suffer this from treating all parts of the body, right? You see these list of side effects on the commercials that go on and on and on, all these adverse effects. And a lot of times, the body can manage it. But with the brain, because of the blood-brain barrier, in order to get enough drug across, because a lot of times maybe only 0.1% of the drug actually gets into the brain, they end up giving kilograms of this stuff to try to get a therapeutic benefit in the brain. And at that level, that's just way too much for the heart and the kidneys and the liver. So with the blood-brain barrier there, you have this need to give a whole bunch of drug. And as you can see, that's going to just have a bad effect where trying to treat other parts of the body, you don't have that issue. And again, the other thing too about the brain is the ways we found to treat it are the vital functions of the brain. You can stop a neuron firing or you could stop the immune system infiltrating, but to do that to the entire brain poses a life-threatening problem. So the brain as an organ, as a whole, is a very, very tricky one to treat relative to all the others. And I don't know how many people realize that there is very, very little for anything disease modifying in the area of CNS.
You mentioned patho-targeting as a core feature of your approach, or rather, patho-targeting is a core feature of your approach. Can you explain, firstly, what patho-targeting is, and secondly, how it ensures that treatments are only activated in disease areas? Because I suppose that's a challenge, right? To ensure that
Yeah.
it targets the right bits.
Exactly, exactly. And it's exactly what it says, patho for pathology, which is, the disease. So it's really, you could say, disease targeting. And we're sensitive, it's fortunate because of the brain is in a privileged space. It's behind this blood-brain barrier. There is no albumin, which is the major protein in our blood. That's filtered out. And because you don't have a lot of these large proteins, specifically albumin, reactions, this chemical reaction can occur. It's not going to happen in the rest of the body because there is a lot of buffering in the blood. But in the brain is there's very little buffering, and you are going to have the pathology when you have the rupturing of the neuron, any sort of the tissue actually rupturing, which will be in the case of a seizure or TBI or Alzheimer's or any of these cases. Those neurons release chemicals into this privileged space. So you kind of have this area where there's not much buffering happening, and you have a lot of high metals and high reactive oxygen species. We found that that's like you could make that mimic that in a test tube, that in that sort of environment, a thiol will oxidize all the way to a sulfonate if it can chelate a metal. And that's very key because this is very specific to the damage and the disease. Only damage and disease is going to have this pathology. And only in this pathology will our drug actually react and become the active molecule. So by being able to target the pathology with really synthesis, this is where I'm going to synthesize my drug inside this pathology. You now have precision that's beyond anything they've been able to accomplish so far today. They they try to do prodrugs where you mask the activity, and then when you get to an environment, say where the pH has changed or it's a tumor, so it's releasing an enzyme at high amounts, this enzyme may cleave off that cap. They've tried that. But what happens is in the body, the cap falls off prematurely, or you don't get enough enzyme that the tumor's making to trim off the cap. It sounds great, but in practice, none of these other approaches to target the area of damage has worked. And we are we are utilizing the actual chemical environment that is so specific for the area, more specific than releasing an enzyme that other cells also release, just maybe not as much, and it's more than just changing the pH because you're in a different part of the cell. Because every cell has that same, you may be able to direct it to the mitochondria, but you're now in all the mitochondria. This is the only way so far has anyone even offered an idea of using pathology to make your drug active and be very precise. And for the brain, it's the only way we're going to be able to tackle these diseases is with this level of precision.
Outstanding. And how does Capton technology leverage reactive oxygen species, ROS, to deliver precision treatment? Or do you feel that's been covered off sufficiently?
Well, I'll clarify it.
That'd be
Um,
great.
yeah, the oxygen in reactive oxygen species is actually going to be incorporated into our Capton and become the drug. So it's utilizing it by it becoming part of its synthesis. It's taking the oxygen. Every thiol is binding up three of these and becoming a sulfonate. And it's that sulfonate that has the therapeutic activity. There's a lot of sulfonates out there, and sulfonates are a thiol with three oxygens. They have a lot of therapeutic benefits, but again, they're highly toxic because they'll do it everywhere in the brain. And sulfonates don't cross membranes well. They're more water loving. They don't really like crossing these lipid areas. But if you take off the oxygens and make it just the thiol, it now has no therapeutic activity. So it's not going to have this effect on healthy things. And it finds its three oxygens back only in this reactive oxygen species sort of soup. And one thing I want to point out, because was known that you don't want to mess around with ROS when you're trying to have a therapeutic. The ROS reactive oxygen species has a huge benefit. It's a signal to the body saying something is wrong. It's the way the body signals for help to come. So if we were to really mop up all the reactive oxygen species, when anything happens, we'd actually be turning off repair. And this would be bad. But it just so happens that the level of reactive oxygen species that's released, say, by running or by just knocking your head or having these events that the body can handle and to signal come help. The chemistry doesn't happen at that level. We don't utilize ROS when there's, say, only a few around, right? Enough to signal damage, come repair, but it's not enough for us to engage. But when you start getting higher levels of these reactive oxygen species that you find in damaged tissue now, you get hundreds now infiltrating the space, thousands, because the tissues, the cells are being lysed or rupturing, opening up, bursting. When you have that much around, then our thiol, then the chemistry actually now works. The kinetics will now happen, and our thiol will engage and pick up three for every one, become the active drug, lowering the ROS in the environment because ROS is also causes damage. You know, it's a happy medium. You want a little to signal come help, but you don't want too much. So we help keep that level at that right level. And once it gets down to, you know, you still have elevated levels, but not pathological levels, the chemistry stops and no more drug will be made. And we don't interfere with the repair, damage repair mechanisms of the cell. And that's something, that's a key point to our technology because I know we've had people say no to us and walk away because of that, but not tell me. So, not give me the chance to explain, look, we are engaging ROS, but not at those levels, only at the really high levels. And again, that's not by design. That's just how this discovery happened to be. So, it makes this so amazing because it's so fitting and so perfect in so many ways.
Well, no, absolutely, and a key point for helping people understand its safety and efficacy.
Yep, exactly.
Excellent. So, what advantages does Capton's thiol-based chemistry offer over other biological drug delivery systems?
For biologics, one thing about them, they're very expensive. They're not that stable. And batch to batch, you could have differences. Um, we're talking with our approach, they're small molecules, which can be very stable. Manufacturing is much easier making a small molecule than trying to make a biologic. Delivery of a small molecule is oral, much easier than having to, say, inject. Um, with a needle or have some sort of a pump installed in your brain. And the cost, of course, is drastically lower, going to be lower with having a small molecule therapy versus having a biological therapy, where the cost is astronomical. So it's very, very exciting that we could have such a therapeutic benefit in a small molecule because of manufacturing cost stability. I mean, to have all that as well really makes this remarkable.
Well, no, you're right. I mean, really sort of ticks all the boxes. Safety, efficacy, and cost probably being the three sort of major things a drug needs to meet.
Correct.
Outstanding. Excellent. No, Sarah, you're doing really well. Brilliant. So now we're going to walk through the different types of, well, the different targets of the drugs. So CPT-021 is your lead program targeting Alzheimer's disease. What makes this therapy different from other treatments being developed in this particular space?
So with CPT-021, this one's unique in the fact that it's currently actually is in the clinic. It was on the market, but pulled for lack of efficacy. The active drug of that molecule is homotaurine. Now, there's a company right now who has it in phase 3. They're about April 1st. They're going to announce the results. Um, we already know the results. No efficacy. It's safe. The safety, of course, because they're dosing at the low enough level before you see toxicity, but there's no efficacy. Um, Again, the reason is you cannot get enough drug at the site needed to have a therapeutic benefit. Right now in neurology, that has been impossible to do. It's either the blood-brain barrier or, you're turning off the entire brain from firing all the neurons. So you can't do that. You can't dose that high with something like that. But with the way we're giving homotaurine, we're not giving homotaurine. It's going to be the thiol analog. Homotaurine is a sulfonate. We give the thiol, which is homocysteamine. That's going to be benign in the body, inert, very low level. But when it engages the Alzheimer's pathology in the brain, it will synthesize and be converted into homotaurine at levels folds higher than what they can do now. Micromolar, we saw 10 micromolar, which is a very high amount in our animal studies, by giving a tiny, tiny, tiny little amount, because as one molecule converts to drug, another thiol will move into that space and replace it. So you get the sink effect, and you can actually build up concentration of drug at site as it's converting. So with our CPT-21, they know homotaurine does have an effect. They see it slightly if they could only get more there. Homotaurine will engage the GABA system, and it silences neurons, not only from firing, but will also silence the immune system. And with our approach, we'll be able to finally deliver the levels needed to engage all those targets at that site of damage to actually stop the immune system from invading.
Phenomenal. And now on to epilepsy. So, CPT 004 takes on refractory epilepsy, an area with few effective options. How does this therapy work to prevent seizures in a way that other drugs can't?
Now, this one is also unique in its own way in the fact that it's not necessarily the sulfonate it becomes, but the process of becoming the sulfonate where it has its most power. Recently, well, it's not so recently now, maybe 6 years ago or so, it was uncovered that preceding any seizure event in 90% of the epilepsy cases, about 90% of types of seizures, there is this huge ROS spike, a spike of the reactive oxygen species that triggers the seizure. And they had done studies where if you could mop up that spike and prevent that spike, you prevent the seizure from occurring. This is now the first hypothesis of a disease modifying to a seizure event. You're actually targeting something that's causing it. Well, the way our Captons, when they engage pathology, is by engaging the ROS, and they engage it really fast and they mop it all up really quick. Now, we've done all of our animal studies was done on O4, and we did prevent a seizure. We sent our candidates to the epilepsy screening program, which was funded by the NIH. All epileptic drugs have gone through their program. You have to pass their bar in order to become an anti-epileptic drug. And we got an email from them that because we sent them 4 samples and only one was O4, the other 3 we knew would not chelate and should not work. They wrote us back with great excitement saying, okay, one of yours has prevented a seizure. Like, how fast can you get us more? We sent them more and it repeated. So we have all the evidence that once converted, but we hypothesize in this case that it was the mopping up of the seizure at such speed and instantaneously almost that prevented the seizure event happening. And if that is the case, then we can address about 90% of the seizure cases out there. And 30% of all cases are resistant to drugs, have some sort of drug resistance. So in this case, you wouldn't have drug resistance with this sort of approach.
Amazing. So strong science backed by real world evidence. It's fantastic. So now on to glioblastoma. A horrifying form of brain cancer and often fatal. CPT GBM is tackling glioblastoma, one of the most aggressive and deadly brain cancers. How does CPT GBM address the challenges of recurrence and residual tumor cells, which I would imagine are more than challenging to remove given their location?
Yes, that is correct. Um, well, two of our candidates that we found are actual chemo drugs. So there are two sulfonates out there with known chemo properties that have a lot of published literature to treating GBM, stopping the cells from growing. Again, it's very toxic because it'll stop all your cells from growing. But two of them, you can make a cap. Two of them fit this approach where if you make the thiol, it will chelate a metal and become the sulfonate in this environment. And the microenvironment around GBM is fitting for our oxidation process. It has all the components needed um, to oxidize into the sulfonate. So now we have found two chemo approaches where you're stopping again the DNA and dividing, you're stopping the cell from actually growing. Um, that can be turned on only when it engages the tumor. GBM is very different in the fact that it grows. It doesn't grow as like a solid mass, as a ball. It grows like a spider web and it sends out all these tentacles. So you have a center mass with all these tentacles that go out and that's why it always comes back. They'll go in and they'll first remove a big chunk. They'll scoop out as much as they can. And then they try with laser therapy and chemo and everything to get all the others. But the places that it reaches, it can, goes around important vessels. So sometimes due to location, they can't surgically remove it all. But you can imagine at the end of all these little tentacles, may just be one or two cells, but that's all it takes, right? Say, you remove the majority of it. You can never get all out at the very fine ends. And in time, those will start growing into their own individual tumors that will then send out, all these tentacles. So you will always get reoccurrence of GBM because you can never get it, remove it all from all these fine ends, the tentacles. With our approach, you know, the tumor cell is going to synthesize the chemo to kill its own self, right? To destroy itself. So in our case, you would remove every one of the cells, and they won't be able to regrow into a tumor. It's a totally different approach. It's a whole new way of thinking about it, and it removes the barriers that they currently have with GBM.
Where are you in the clinical development process for your three lead programs, and what's the timeline for clinical trials?
Yes, so we're in the same place, you could say, with all of our candidates. We pick our candidates based on whether there is preclinical evidence showing that the sulfonate will have a therapeutic benefit. And then we look and say, if this was the thiol, would it have the right properties to undergo the right chemistry and become the sulfonate and damage? If yes, then it becomes one of our candidates. So right away, all of our candidates already have all the preclinical evidence. We don't need to worry about the PK of the drug because we're not giving the drug. So every candidate is just the same distance. I'd say about 1 year because all of these being highly unmet, with high unmet needs, they will qualify for fast track designation, which just means the FDA will look at it sooner and will, you get put up at the front of the list. They're all about 1 year away. Every one of them, what they need done is the thiol needs to be made um, because of the dose, the level of dosing, and we know a lot about thiols as drugs. We are confident that there won't be a toxicity issue. And if there are ways to handle it, such as liposomes and things. There's a lot of things you can do. So we're not worried about there being a toxicity issue. You can't go any further. It would just be we need to formulate this a different way. But they would need to be the material made, formulated, um, and that material run on a tox studies done, and that's it. Then they're all ready to go to test in human and see, does that sulfonate as preclinical data show? Because a lot of the problem in translating from preclinical to clinical, again, is from rodent to man, is it got in the brain in the rodent, but it doesn't get in the brain in the man. Again, you can't, and however it got in rodent, it's not the same in man. And again, we don't have that issue. We do know that these styles and the size of them, it is predicted. We do know they will cross the membrane. Again, the chemistry is highly understood. We do know it will become the sulfonate in this environment. So then the question comes down to, okay, if you engage this target, do we really have this benefit in them? So, but all of them, not just the three. So anytime we find a candidate, they're all going to just have this about this year step. There's no efficacy to do, you see, with the new candidate because it was found based on already strong data in the clinic showing that there will be a therapeutic benefit if you could get it there at the site of where it's needed at the right concentrations.
Well, absolutely. And because, as you say, you understand it thoroughly, you know precisely how it'll behave.
Yeah, exactly.
Outstanding. Okay, great. So, fast track designation. So, firstly, I mean, the name is fairly self-explanatory, but people probably don't know what it means in a pharmaceutical context. So, what does fast track designation mean for enabled therapeutics? And how could it accelerate the availability of your therapies to patients, which is the important thing, isn't it? Getting the therapy to the people who need it.
Yeah. It would definitely speed it up by, a few years. Fast track to the FDA means, instead of it taking 9 to 12 months to get back to you, they have to give you a response within 3 months. It limits the time. So and it's good also to tell investors, we'll know by this date, the FDA has to be back in touch with us with a decision by this certain amount of time. And each step you could have fast track designation for filing the IND and then for filing your phase one data to move into phase two at all those steps. And so if you could, bring down the timeline by 9 to 12 months each time, 6 to 9 months each time, you could really shave off a few years to getting it to patients.
Amazing. And where are we? So, strategic licensing partners. So, we're going to talk about collaborations next. So, you're very much seeking strategic licensing partners. So, what kind of collaborations are you looking for? And importantly, how can partners get involved?
So our goal is just to move this technology forward. Whether we do it or someone else does it, it doesn't matter. It needs to go forward. This is real, that is big, and it's going to change a lot for good. So we're open to any type of partners, whether it's in form of funding, and we commission the CROs to do the next step, IND enabling and the phase one trial, or a partner who wants to take it in-house and they perform these next steps themselves. Either way, we're open to whatever it means to move it forward.
No, and I love that. I mean, it's not about gatekeeping the technology. It's about, true scientific collaboration so that
I'd give it,
disease can be cured. That's fantastic.
I would give it 100% up if it meant if that was the only way to go forward, because
Amazing.
it's
Amazing.
not about me. I'm
Yeah.
I was just, a vessel that found this. It has to get it out.
Absolutely. Yeah, fantastic. Okay, great. So, how important are partnerships in terms of scaling and commercialization of Capton technology, particularly when trying to reach global markets?
Incredibly important. It is not going to go anywhere if we don't partner at this stage. We managed to do all the preclinical work ourselves, doesn't cost much. We managed to raise 6 million among friends and family and ourselves. And you can do a lot of this stuff turned inward, a lot of, gathering the data and all that. But now at this stage where we have to go into human trials and now get it out to the people, it requires 100% partners and people would go way beyond us and are experienced at everything that we are not. We are very good at bench work. I myself am a bench scientist. I'm not an executive by trade. I've become a CEO out of necessity to move this forward. But there's a lot of steps needed now that go beyond our expertise. So we really do need the partners in drug manufacturing, in running the clinical trials, in the funding, and in just getting it out, then marketing it and manufacturing it and getting it out to people.
Absolutely, and I mean it's an important point to make because I would imagine the majority of people aren't aware of the many moving and necessary parts of the process.
Yeah, there's a lot. Yeah.
Excellent. Well, beautifully explained. So, okay, great. So, beyond your current pipeline, where do you see the greatest opportunities to expand the application of Capton technology in neurological disease? So, what does the future hold in terms of expanding the technology's use?
Well, I'm very eager to find out myself, and I say that because this is just what we've come up with by uncovering the idea that thiols will oxidize. You can get sulfonates made by pathology. Just that bit of knowledge isn't known. That was what we discovered. That's a huge discovery, and we haven't published on it yet. We have an IP, and because of IP and all this stuff, we were advised not to publish yet. So it's not yet been told that this can happen. Once it's known and all the experts are able to think about this, the medicinal chemists, I'm very excited to see what they are going to come up with and be able to do. I mean, on my own, I can see you can have a molecule, there's a sulfonate that's actually the color brown and it will turn brown among oxidative stress. What is that? That's a hair dye. That's something where I'm thinking, it's peroxide that makes our hair go gray. But imagine if, when your hair gets that oxidative stress and starts dying, it actually converts a molecule of brown and recolors your hair. That's way far out there, has nothing to do with neurological, but it's just showing that there are so many things you can start doing. If you can make a color, you could have cancer turn a certain color maybe on your skin, and it could be a diagnostic. The other things you can think of, other privileged spaces beyond neurology, are the joints. You hear of the blood-brain barrier. There is a joint barrier, and albumin does not get inside, the synovial space. There is a chance that maybe this technology can be used for arthritis and gout and a lot of things with the joints. So what I like to say is we've only just found like the tip of an iceberg with this thiol-based patho-targeting sort of redox chemistry. It's a huge iceberg that, once we get this out and people can start thinking on it and realize this process is occurring inside pathology, it's going to go places I can't possibly imagine. I know that.
It's amazing, and I mean we've done many pieces on novel therapies, and it's always remarkable the sheer potential for expanding their usage, and the multiple uses they often have. So wonderful. So what's your message to patients, families, and healthcare professionals who are hoping for new solutions in neurodegenerative and neurological care? Because I suppose that's the human element, isn't it? Sort of beyond the science and the distribution. What would be your message to people, affected by these conditions, or people who know have loved ones who are affected by these conditions?
I'd like to say that there is an answer that in just in a few years, because with AI and everything, we can't be the only ones who've uncovered this. This is going to be found by others too at some point. I'm pretty much sure of it. So even if we don't succeed at getting it forward ourselves, I have a good feeling AI is going to figure it out and come to us, and that there is hope. A lot is known. There's just a couple little things that we need to solve, and they've been solved. And just hang in there, because there is a future where neurology is no longer the weakest branch of medicine. It can be the strongest branch of medicine.
Outstanding. A very moving note to end on. And just before we conclude the recording, where can listeners go for more information, please, Sarah?
So they can go to our website, which is at www.enabledtherapeutics.com, or you can also, DM me, find me. We're LinkedIn at Enabled Therapeutics, or also my name's Sara Isbell. on LinkedIn, and I would love to continue the dialogue with anyone interested.
Sara Isbell, CEO and co-founder of Enabled Therapeutics, thank you so much for your time today.
Thank you.